We were here last time, so let's continue. So here we are talking about the direct examination of the specimen. What do I mean by direct examination? It means that you are not making the culture, but you are rather taking the specimen directly, like CSF or blood or urine, and making a gram stain of this specimen and then viewing it under the microscope. As we did yesterday in the lab, and as we explained in the last lecture, that in some specimen like CSF, if you see one bacilli, let's say gram-negative bacilli or gram-positive cocci, then this is very important because these are sterile sites which should not contain any bacteria. So in case you see it, you have to report directly to the physician so the physician can start the treatment course. That's what I mean by smear of primary specimen or direct smearing. Making a smear of the clinical sample itself, not the culture. The most common is gram stain. This is really the most common and widely used. Then we have acid-fast stain. We have the cold, the Kenyan, and the Thiel-Nielsen, this one here. We will have in the lab acid-fast stain, and we'll talk about this. This is mainly for MTB, Mycobacterium tuberculosis. Some parasites like Cryptosporidium mainly, or Isospora belli. But this stain, acid-fast, goes for the MTB, Mycobacterium tuberculosis. KOH. Of course, back here, acid-fast from primary specimen, we usually make acid-fast stain from the sputum directly. And under the microscope, you will see the blue background because we have methylene blue here, and you will see the red bacilli because it's carbulfoxin. Then we have KOH. KOH, potassium hydroxide, this is mainly for fungus, ringworm and others. Tinea worm also, not the tinea parasite, but the tinea fungus. Tinea rubrum, tinea angium, tinea capitis in the head. So KOH is mainly for the fungus. And what does the KOH make? It dissolves the keratin, which might mask the fungus. So the keratin is dissolved and the fungus is seen as fluorescent green. Less commonly used stains, oramine rhodamine, PAS, periodic acid Schiff, calcoflower white, also for fungus. These are less commonly used. This is the most common. Then comes this, and then comes this for the fungus, mycology. Let's go to the most important thing here, which is the media, culture media. We have culture media, and we'll talk about this in the lab. We have the all-purpose media, which we call nutritive media. It's a media for everything. It's not selective. It supports the growth of all bacteria, gram-positive, gram-negative. They all grow on blood. And not only that, sometimes you see fungus, yeast, also grows on blood. So this is a nutritive media, non-supportive, and non-selective, and all-purpose use. Chocolate is like blood, and even more. Chocolate can support the Haemophilus influenzae bacteria, where blood cannot. So chocolate is even wider than the blood. And usually blood is SPA, 5% sheep blood agar. Nowadays, they are using horse also. SBA, sheep blood agar, is a nutritive and differential. Comes from the idea that it displays hemolysis differently. Some bacteria, they eat all the blood 100%, leaving the blood colorless, almost colorless, if it's strong hemolysis. Some bacteria, they eat part of the blood, not all of it, leaving the blood agar green, a greenish discoloration. And some bacteria do not eat the blood, so leaving no hemolysis at all, zero hemolysis. Or this hemolysis display pattern we call blood sometimes as differential media. We have selective media, the media that supports the growth of one bacteria, one group, and inhibits another. And we talked about this in the basic micro. We said that McConkey supports gram-negative, but you do not expect gram-positive to grow on McConkey. Gram-positive is inhibited either by the salts and the crystal violet in the McConkey. So McConkey is selective media. It supports one group of bacteria, but also inhibits the other group. Because of this, we call it selective. It selects one group. It's differential. It can be differential. How? This media contains pH indicator, and this pH indicator is called a neutral red. And the bacteria can produce, after the growth, it produces acids as part of the waste. And these acids, they change this pH indicator from light red into dark pink, like E. coli. So this change in color makes us describe this media as differential. So any media where you have change in color, then we call it differential. So media can be selective, and media at the same time, it can be differential, like McConkey. So McConkey is a selective differential media. And then we have enrichment media. A media supports and boosts the growth of some bacteria and inhibits another. Some bacteria, like for instance in stool, you have only small amounts of bacteria, and you have lots of microbiota. What does this media do? Inhibits the microbiota, and at the same time, this small amount of pathogenic bacteria, it will boost its growth and multiply, which will multiply within 6 hours. After 6 hours, you take one drop from this media and spread it on McConkey or blood. Before this, if you spread it on McConkey, it will be negative because it's a very small amount and can be masked by microbiota. But after 6 hours, thioglycolate or BHI, brain heart infusion, this small amount of bacteria will become enormous. The count becomes very high, and then you can see it next day. If you just subculture it here, you can see it on McConkey easily. Okay, so we have 3 types of media: nutritive, selective, and differential. Selective and differential, or selective or differential, because some media, they are only selective. Some media, they are only differential, and some media like McConkey, selective and differential together, and enrichment media. Okay, we have 3 types of media, and I think we talked about this in details in the basic micro. But also we will continue talking about these media in details in this course also. And this is what I mean when I said that blood agar is a differential media. This is what I mean. Some media, of course, on 5% sheep blood agar. Human blood, it will not display the hemolysis thing. So if you look at the one in the middle, you will see that the bacteria here has eaten most of the blood. See, there is no blood here. And sometimes if the bacteria is very strong and the blood is very fresh, you will see this area not yellowish as it looks here. You will see it totally colorless. Okay? This is what we call beta hemolytic. The strongest form of hemolysis is called beta hemolysis. Then this bacteria here, this plate here, if you see it's light green, we call this green discoloration. This light green comes from bacteria that partially eats blood. Not fully, not completely, partially. Example of this is Streptococcus pneumoniae. An example of beta is Streptococcus pyogenes. And then gamma. Gamma, very weak or sometimes no hemolysis at all, like enterococci. So we have alpha, beta, gamma. The highest, the complete hemolysis goes for beta. Partial goes for alpha. No hemolysis or very slight goes for gamma. And for this reason, we call 5% SPA as differential media, which is originally supportive and all-purpose use. Look here, McConkey. On this side here, it's pinky. Why? Because the bacteria, which might be Klebsiella pneumoniae or might be Escherichia coli, it has utilized lactose. And after utilizing lactose, what happened? Acid is produced. And this acid has changed the pH indicator, which is a neutral red, from yellowish or colorless into pinky. While this bacteria here, it did not ferment lactose, and the color remained as it is. For this reason, because of changing color, McConkey is differential. And because it doesn't support the gram-positive cocci, like Staph, we call it selective media. So McConkey is selective plus differential. Specimen preservation. Specimen preparation. In some cases, you have to process the specimen before working with it. You have to process it. This processing of the specimen exposes the bacteria and maybe gets rid of microbiota and also makes the numbers higher and higher. Example is homogenization. Homogenization, grinding and mincing of tissue. If you want to culture a tissue, you have to put it in in a sterile saline or bone sometimes. I remember we did culture in bone. And what we did is that we brought a crucible, more crucible with mortar, pestle and mortar, sterile, autoclaved. We added on the bone, small piece of bone, we added sterile saline, and then we started manually grinding and grinding. So grind until the bone became just like a fluid or like a suspension. And then we took 100 microliters of this suspension or 1 drop and spread it on blood agar. See what we did? Grinding and mincing, tissue and bone. Concentration, centrifugation and pelleting, like in CSF. Normally the CSF is colorless. Sometimes it's lightly turbid. So you have to centrifuge to make a small sediment or pellet, and then take from this pellet using sterile swab or using a sterile inoculating loop and spread on the blood agar or McConkey. So we call this concentration and sedimentation. Sometimes you have to make filtration. You have large volumes of pleural fluid or ascitic fluid, and you make filtration, and you take the filtrate and make a culture out of the filtrate. Or sometimes you can use also the fluid which has been filtrated for something else. Filtration. Decontamination. Respiratory specimen for MTB, mycobacterial tuberculosis, and Legionella. Legionella pneumophilia. It's a bacteria that causes pneumonia. So we have to decontaminate. We have to remove all microbiota, leaving only the Legionella. Decontamination. Mixing and vortexing. For traditional swabs, 1 ml saline in vortex for 20 seconds. What we do is here, I'll give you an example, is that we have, let's say we take a swab, high vaginal swab, and then we suspect this lady of having trichomoniasis, Trichomonas vaginalis. What we do, sterile saline, add it to the swab, put it on the bench for 15 minutes, and after this, take the 1 ml, and what do? You sediment, you centrifuge, and you take the sediment, you vortex up and decant the fluid, take the sediment, vortex. Vortex means you have to dislodge, disturb the sediment, and take a drop and under the microscope to look for the Trichomonas vaginalis. So this is mixing and vortexing. We use this for high vaginal swab. So these steps, let's say pre-testing steps. Before you test, you do these, either homogenization, concentration, filtration, decontamination, or mixing and vortexing. These here pre-testing procedures that prepare and make the specimen ready for culture and ready for examination. Inoculation on solid media. Look, this is what we call inoculating lube. We commonly use two types, either the one with 10 microliters or the one with 1 microliter. I prefer this one because it makes it easy, especially in urine, because it makes it easier for us to make the count. Both will do. So quantitative inoculation in urine culture, especially in urine culture and sometimes burn tissue culture, but urine is the most common. You have to do what? You have to do quantitative inoculation, which is only add 1 microliter. You dip this inoculating lube vertically in the urine one time and out and spread streak on the surface of this plate. 24 hours later, you have to count the number of colonies. You have to count them. So the urine culture is the only one that we count to make quantitation. Burn tissue, yes, it might be, but not very common. Sometimes we have semi-quantitative inoculation, seeking for inoculation and identification. Also, specimen can be diluted by successive streaking, like this one. Look, this is primary streak, so it's very concentrated. Then you take the secondary streak. If you see this is less dense than this, then the tertiary, less dense, and the quaternary, number 4, you can see isolated colonies here, which you don't see here. So this dilution made what? Made the difference by giving you isolated colonies. We call this quadrant streaking. Quadrant means 1/4. Quadrant, 25%, 1/4. Quadrant streaking. 1, 2, 3, 4. Primary, secondary, tertiary, quaternary, quadrant. Okay, now let's come to something which is also important, which is the oxygen requirements. The oxygen requirements. How much oxygen we need for the bacteria? Of course, the bacteria needs this oxygen. Aerobic bacteria, which depends on oxygen only, it needs not 100% oxygen. You might be surprised, not 100%, 21%. But there's 21 oxygen, like Escherichia coli. CO2 very minimal, 0.03. Anaerobes or anaerobic bacteria, it's the other way around. 0 oxygen, no oxygen at all, because the oxygen kills the bacteria. We have hydrogen, we have 10% CO2 for the anaerobes, and also there is nitrogen, 80 to 90, like the normal atmosphere, but without oxygen. Example, Clostridium botulinum. This bacteria only lives where there is no oxygen. Clostridium botulinum. Comment, we have also jar. So this anaerobic, we have what we call anaerobic jar. Sometimes there's a bag, anaerobic bag, or a chamber. Of course, we have a sachet, and this sachet or sachet, it contains a reaction, biochemical reaction, that sucks up all the oxygen. We will have this in the lab. Capnophils or capnophilic conditions. Capnophilic means something related to CO2, carbon dioxide. Capnophilic goes here. It means that we have 10% CO2. So capnophilic bacteria means CO2, 5 to 10%. How can we get this? By candle jar. You know the candle jar? There is the candle. You put your plates in the jar, put candle, light it on, and close. The candle, after less than a minute, it will die out and it will be off. And the candle will release some CO2, which the bacteria will use. Example of this, Haemophilus influenzae. Example of this, Neisseria gonorrhoeae. And other bacteria like staph, strep, they prefer capnophilic conditions. Although they can be facultative anaerobes, they can this or that, they can live with oxygen or without, but they prefer the anaerobically or even capnophilic. so that some of the characteristics you can see easier, like hemolysis. Hemolysis is much better with capnophilics and even anaerobic. Sometimes you can see the bacteria, the morphology of the colony much better with capnophilic conditions or even anaerobic conditions for staph. Although staph is facultative, it can live with or without oxygen. Microaerophils, which there is oxygen, but in a very small amount. Full oxygen, 21%. Microaerophilic, which is very small amount of oxygen, 5 to 10%, almost half. And 8 to 10 CO2, like this. Example is Campylobacter jejuni and Helicobacter pylori. These two here, they are microaerophils. They like oxygen, but in small amounts. They are not greedy, and they are modest bacteria, not very much high requirements. And of course, every time we have to have a jar and a bag. Those bacteria that need special conditions, other than facultative anaerobes and aerobes, you need to have jars, bags, chambers, a system that can contain the blood cultures. Okay. What organisms to look for and to report? Of course, in the CSF, you have to report every organism. In the pleural fluid, the same story. Ascitic fluid, also the same. Why? Because these are sterile fluids. And these fluids, they should contain zero bacteria, no bacteria. And if you find this bacteria in these places, you have to report them, regardless of what type. However, in some places, like let's say urine, or like let's say mainly stool, sputum, anything that is exposed and has microbiota there, you have to be careful, because in stool you will never get any culture negative. All the time there is bacteria. So you have to choose and identify the correct bacteria that is pathogenic bacteria. And this is the challenge for microbiologists, is to tell which is the pathogenic bacteria and which is the microbiota. And sputum, the same story. Panic values. Panic means afraid. Somebody with, let's say, FBS, fasting blood glucose, 600. This is panic value. Or somebody with glucose, 60, panic value. These numbers and these figures, most of the time, they are incompatible with life. You cannot find anybody with glucose 40, because then he will have died or at least in coma. Therefore, in microbiology, we have the same. We have 5 or 10 panic values. Let's start with the first one. Positive blood culture. Any blood culture, then you have to be panicked and you have to take it priority, give it priority. Because blood is usually sterile. And when the culture in blood is positive, then there is a problem. 2, positive CSF. Again, CSF is like blood. Zero bacteria. No organisms there at all. No microbiota and nothing. If your microbiota in your body, which is normal, reaches your brain, it will cause big problem for you. So positive CSF is another panic value. 3, S. pyogenes in surgical wound. Streptococcus pyogenes. Streptococcal pyogenes in wounds, they will cause a dangerous disease called necrotizing fasciitis, where this bacteria starts eating your flesh, the meat of the human being, the flesh of the human being. We call it necrotizing fasciitis. So if you have S. pyogenes in a surgical wound after an operation, then this is a big panic value, and you should call the physician. Gram stain. You have seen Clostridium perfringens, and Clostridium perfringens causes gas gangrene. Your tissue becomes black, and when your tissue becomes black, it means your tissue is non-viable, dead tissue. And the solution in this case, amputation, cutting the leg, cutting the hand, amputation. So when the gram stain shows Clostridium perfringens, you have to call the physician. Positive smear for Plasmodium. This is malaria. Malaria is a dangerous disease. That's in parasitology. And if you see rings in the red blood cells, if you see rings in the red blood cells in a Giemsa stain smear from blood of a human being who had shown fever and was, let's say, in Africa, traveling to Africa, then this is a panic value and the physician should be called. Positive acid-fast smear. We said that the acid-fast stain means that we have MTB, Mycobacterium tuberculosis. If you see red bacilli on a blue background, then this is Mycobacterium tuberculosis. You have to call the physician and tell him or her that we have MTB case, highly suspected. Number 7, positive cryptococcal test. Cryptococcus neoformans. This is a fungus and mainly causes meningitis. So if you see this test, cryptococcal test, like quelling test and other tests, then you have to call the physician. Detect significant pathogen. So you have a list in your country, and this list in each country has its own list, and you have a list in your country provided to you by MOH, and every time you have any bacteria from this list, then you have to call the Ministry of Health. Each country has its own list of reportable bacteria or reportable pathogens. We call them reportable or sometimes notifiable. So we have these 7 or 8 cases where the physician should be called. And don't wait for the culture results to come out after 3 days. This way, we have finished this chapter. And let's start with a new chapter, which is Staphylococcus species. Chapter 13, Diagnostic Microbiology. By the end of this chapter, or the following objectives should be clear to you. First, describe characteristics of what? Of gram-positive cocci that is catalase positive. This chapter, which is chapter 13, it's about catalase positive, gram-positive cocci. Describe characteristics of this. Correlate culture and microscopic characteristics. Make gram stain, look under the microscope, direct wet mount maybe, and make correlation. Or let's say, make sediment for the urine. You see lots of bacteria, most probably you will get positive culture. So you have to make correlations. And these correlations are very nice because it means that you are aware of what's going on. Describe culture media as we did just minutes ago. So here we have culture media, but for the bacteria itself, the catalase positive, the gram-positive cocci. Explain principle of coagulase test. Why coagulase? Because coagulase test is the test that is usually used to detect Staphylococcus aureus, the most pathogenic of all in the staph. Staph, we have many, but the one that is really important is the Staph aureus. And how do we tell this is Staph aureus or not? A very common test is coagulase test. And also we'll talk about it in the lecture, and not only that, but also in the lab. List associated diseases. What are the diseases that staph causes? Staphylococcus aureus causes a list of diseases. What are they? We'll talk about them in this course, in this chapter. Outline biochemical tests for diagnosis. Of course, you can never make diagnosis, definite diagnosis, confirmed diagnosis using only one test. You have to use multiple tests, sometimes 1, 2, 3, 4, 5. The more tests you use, the more you are certain about your results, generally. Of course, there is a limit for this. You cannot use 10 tests just for things that you can get it with only 2 tests. Define MRSA, HAMRSA, CAMRSA, and LAMRSA. What are these? MRSA is methicillin-resistant Staphylococcus aureus. It's a bacteria that is Staph aureus, but it resists many antibiotics, which means this is a dangerous bacteria, a dangerous species or a dangerous variant of the Staph aureus, which we all don't like to have. Look at this list. This is a list of all staphs and related species. If you look here, we have the one in red, we have the ones in green, and the ones in gray. The one in red is the most important. Why? Because it's the most pathogenic and very commonly isolated. We call it Staphylococcus aureus. This is coagulase positive, catalase positive. When you make gram stain, it's gram positive. It is cocci. You make catalase, it's positive. Then after this, you make coagulase, it's positive. So this is very common and coagulase positive. There are also staphylococci, but they are not coagulase positive. They are coagulase negative. And mainly these are normal flora, or they don't make any, or they don't cause any serious diseases in special circumstances like immunocompromised patients. Example is epidermidis, Staphylococcus epidermidis. This you have to know because it's a normal flora or it's a microbiota on human skin. So any one of us, if you make just culture from the skin, you will get epidermidis, and it's normal. Hemolyticus, saprophyticus. This is not normal, saprophyticus, and not very common. Logodonensis, Schleiferi, and others. Of course, I am not going to ask you about all of these. The ones that you should have in mind all the time: Staphylococcus aureus, epidermidis, and saprophyticus. These are the ones you should know. The rest, it's good to know them and to look at them and read them. We have also coagulase negative staphylococci, but they are not very common. Hyacus, chromogenes, capitis, yeah, and so on. All of these. But are they important? Not very much. We have also the green one, good to know, like these here, micrococcus. Why? Because it's catalase positive and gram positive cocci. The cocci here is larger than these, and usually this exists in the form of tetras, 4 cocci together, and we call it micrococcus. It doesn't cause any problems, usually. It does not cause any problems to a human being. Micrococcus species like luteus and regenerate. Aleucoccus, not very much important. So I need from you to know this, very important, and also to have an idea about epidermidis, saprophyticus, and micrococcus. If you know these 4, the rest just for you to read. Okay, epidemiology. Where do we find the staph? Well, the staph in some people, it can be on the skin, staphylococci, and normal microbiota. And we call these colonizers. Many people, they don't have the staph at all, but some they have it. And those who have it, we call them colonizers. They have the Staphylococcus aureus colonized on their skin. And we have 3 types of colonizers: either persistent, intermittent, and non-carriers. Persistent, all the time the staph is on their skin. These are people who have the staph on their skin forever, every time, permanently. And they harbor it for a long period of time. Intermittent, sometimes. It could be because you are working today in something, and the whole week you are doing the same thing, let's say in a factory. Then during that period, you have staph around, and the staph is colonizing your skin. If you go back home and forget about the work, then it might go away. So these are intermittent. It comes and goes. And then we have non-carriers, people who do not have it. They don't have, so persistent, intermittent, and non-carriers. S. aureus, Staphylococcus aureus, the most pathogenic. You can be surprised if I tell you that the medical team, 15% of the medical team, almost 10 to 15% of the medical team they have this Staphylococcus aureus as microbiota. Can you imagine? Staph aureus, which causes problem to us humans, some medical team, because they work in hospitals, it colonizes their external nares inside their mouth and the throat nares, and it becomes normal. And these people who have S. aureus as microbiota, they can infect the others, like us who don't have it. This is the external nares, anterior nares, yeah, and nasal cavity, nasopharynx. This is the nasopharynx of the human being, mouth, and this is the nasal cavity. And you have bacteria, Staphylococci in general. You have it inside here. You have it in the back, the nasopharynx, and you have it on the skin. See, on the skin here. And it can cause lots of diseases for skin, Staph aureus, impetigo, cellulitis. We'll talk about them in the next few slides. Aureus means it's gold. Aureus in Greek, I think, it's gold. And staph means grape-like, clusters. So Staphylococcus, clusters of balls, yes, clusters of balls, spheres, that are golden in color. On the skin, inside the anterior nares, and nasopharynx in the back of your throat. If you look here, the epidemiology, if you look here, I have just made this red. This is red. And if you look here, the habitat, normal microbiota. S. aureus, normal microbiota, but not for everyone. Only for certain people who are medical team and 10 to 15%, not more. And it's found in the anterior nares, nasopharynx, perianal area next to in the anus, skin, mucosa, and mode of transmission. The mode of transmission is endogenous strain. Endogenous, it means that some people, these 15% of the medical team, they have this S. aureus as inside. It's coming from within. That's why we call it endogenous. Or trauma. You have a car accident and then it came to you. Or direct contact by fomites, like scissors, like nails, like forceps in the hospital. Or person-to-person contact, hugging, shaking hands. Indirect contact, aerosolized. Maybe sneezing, somebody who has S. aureus, he's or she's sneezing next to you, and all the S. aureus comes on your hands as a mist, so you don't take note, and then you are infected. So most important is the S. aureus. The rest here, we will talk about them in the next lecture. So that's all for today. And if you have any questions, I am ready to answer. Thank you very much and have a nice time.